Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Mehdi Soleymani-Goloujeh. We're going to be talking about extracellular vesicles in regenerative medicine. Mehdi is part of the website known as modernmedlab.com. So again, that's what we're going to be talking about. So Mehdi, thank you for coming. How are you doing?
Hello, thank you, Richard Jacobs, and I appreciate the organizers of this meeting, especially futuretechhealth.com. Due to my invitation, I am here and glad to meet you, everyone. Let's talk about myself in brief, then I will explain my research and interests, findings, my background, future prospects, and what's going on.
Yeah, go ahead. Tell me what part of extracellular vesicles are you researching?
Yeah. Before anything, I will explain about myself in brief, then I will explain my research areas.
Okay, go ahead.
I'm Mehdi Soleymani-Goloujeh, PhD candidate in cell drug and applied cell sciences at Royan Institute for Stem Cell Biology and Technology. Royan Institute is one of the most important and well-known research centers in Iran, as well as Middle East. I'm a researcher who works on type 1 diabetes and seeks the role of extracellular vesicles or exosomes in immune-related disorders. The question is, what are EVs? So what do they do? Actually, EVs are bilayered vesicles produced by almost all cells. Nowadays, it has been demonstrated that cells from different organisms, from eukaryotes to prokaryotes, can release vesicular bodies into the extracellular environment. They were not considered as vital parameters in cell-to-cell communications and other procedures in the past years. Nobody knew their functions and duties in the body, but step by step, and when scientists were getting more information during these years, EVs were announced as biomarkers for a wide spectrum of diseases, or as therapeutic agents to treat diseases. Extracellular vesicles were initially known as cellular waste carrier, while recent studies demonstrate that extracellular vesicles play important biological roles in all aspects of life, from single cells to mammalians. More experiments pave the way of thinking better than before about the vital functions of these vesicles. EVs are considered as significant factors in inflammation and immune responses, antigen presentation, cancer progression and metastasis, immune modulation, coagulation, tissue regeneration, organ repair, cell-to-cell communication, senescence, proliferation, and differentiation in the body. Therefore, a significant attention has been emerged. Since a decade ago, different groups initiated sophisticated research on identity, functions, potential capabilities, applications, blah, 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 of these interesting packages. EV is a collective term which refers to heterogeneous cell-secreted structures comprised of bilayer phospholipid membrane surrounding a wide range of macromolecules, including proteins, lipids, and nucleic acids.
Okay, so Mehdi, there's a lot. Can we focus in on your research? It's how the immune system uses extracellular vesicles, or how the immune system responds to extracellular vesicles from other cells?
The interest in my country between scientists in medicine and biology led them to consider extracellular vesicles, or EVs, as important factors in their research works, and published more related data about isolation, purification, and application of these promising factors. Several years ago, a group called EV Group was established in Royan by Dr. Faiz Shekari, Professor Hossein Baharwand, and their colleagues. Me and my colleagues are working on EVs, and in a related research topic in Royan is covered by aforementioned group. Immune-related disorders like autoimmune disease is one of the most common diseases in developing countries due to well-being and improving community health. I started my own research about type 1 diabetes several years ago. The effects of stem cell-derived EVs in modulating immune system or their effects on regeneration of different tissues are considered to be studied more and in detail, absolutely, in type 1 diabetes mellitus. So
how specifically do EVs play a role in type 1 diabetes? When do you see them, and what type of cells are giving them off?
The most important and the best motivation for me in this field is helping people with these abnormal problems. Also, their families who are involved in this situation. I got interested in making smile on the lips of kids who are affected by type 1 diabetes. It's a great idea, or the biggest dream for any researcher in medicine and biology to treat all diseases. Even reaching this goal is a little bit difficult or seems impossible right now, but I believe that one day everything will be right. Reaching this goal is not far away. As a researcher who knows how the science would be cruel, I emphasize that the sword is double-edged. It's up to us how to use it to solve these intricate and complex problems. I'm an optimist. Therefore, I hope we can pass through these difficulties as soon as possible, particularly by using EVs as biomarkers for diseases like type 1 diabetes, prognosis, and diagnosis, or using them as drug delivery vehicles, therapeutics to facilitate treatment of disease.
So how, for instance, do pancreatic beta cells use EVs? Do they use them? Do they make insulin and then they excrete the insulin through EVs, or like what mechanisms have you figured out, particularly how the pancreas uses EVs?
Maybe one of the options on the table is a delivery of insulin to the people who have type 1 diabetes, but it's not our goal, our dream. Today, we have the options on the table to treating diabetes type 1 with immunomodulating, modulating the immune system, as well as regenerating insulin-producing cell that we call beta cells. And it's our final goal to regenerate insulin-producing cells in the body and modulating immune system cells, and maybe treating type 1 diabetes in this people.
Well, right now insulin is delivered just in a small molecule form. Is there any point in packaging up insulin inside of vesicles to deliver? Or, how would this affect the delivery of the drug itself?
One of the most important things, by virtue of EV engineering, we might be able to modify the contents of EVs by adding therapeutic drugs like insulin or other factors, small molecules or compounds, to enhance their regenerative potential or, their therapeutic effects to delivery of molecules like insulin to the body of patients with type 1 diabetes. It's a little bit difficult nowadays, but by using some technologies like biomaterials and technologies who developed in the field of regenerative medicine, we can reach this goal in near future.
Okay, but what kind of signaling is going on? What kind of signaling involves EVs in the pancreas, in pancreatic cells and in beta cells, in healthy individuals or in sick individuals? You figured out what kind of signaling is going on and what is inside of the EVs that are excreted by cells in the pancreas?
It's a little bit difficult to saying what is the exact mechanism for releasing extracellular vesicles and modifying or manipulating some signaling pathways during releasing exosomes or regenerating beta cells in islets of Langerhans. But yes, there are more sophisticated pathways, signaling pathways that regulate this action, and the mechanism of action is not truly elucidated nowadays.
Well, what are you focusing on in particular? What do you want to figure out about type 1 diabetes? Is it prognosis? Is it early discovery? What are you looking to figure out?
Let me speak about a topic. I believe in the power of nature. In my opinion, we have to mimic the mother of nature to realize what is true or what is wrong. Let's explain more. We all learn a wide variety of things from animals and creatures around us. These are known as instincts. Eating, drinking, running, hunting, going to the shelter, defending, etc. are most dominated actions we do. We learn most of them in ancient times from whom? It was not the nature, accidentally. Yet, our mother nature trained us in some aspects. So why should not we use our body features to get rid of some of all disorders? Does it possible or not? I think we have neglected this important point in our life. Good decision making can play an important role in managing this hypothesis. We modify or manipulate some chemical drugs to treat disease. I don't know. Tablets, syrups, ampoules, etc. These are very useful, but whether it is rational to getting back and investigate our body's abilities when confront or face dangerous situations or not. Nowadays, we are talking about stem cells. Do we produce these types of cells artificially in the labs? Absolutely no. We find out and extract them from our body. We just isolate them, expand them, proliferate them, and use them in different indications. This is a simple example of utilizing our potential abilities to manage discomforts and disability. Generally, any type of cells can produce EVs. However, these cells can be one of the problematic cells, and we can use their EVs to diagnose the cause of malfunction or disease. So, extracellular vesicles can be used as some indicators of our body if we are we are sick or not, or we are a healthy person. So extracellular vesicles can use as diagnostic factors, and even we can use them as therapeutic factors to treat some diseases like type 1 diabetes.
Well, how specifically would they be used to treat type 1 diabetes? Right now, we give people insulin, but would you, what would be different with using EVs?
Extracellular vesicles or EVs derived from stem cells, for example, mesenchymal stromal cells, are valuable candidates for induction of regeneration as well as immunomodulation. The desired goal is regeneration of tissue or organ. Now we have some communication factors, extracellular vesicles, which facilitate this. Gathering some cells like macrophages is conducted through this pathway beside others. Then we have, we solve the problem easily, just modulating immune system, and besides it, we can regenerate insulin producing cells. Then the balance we can reach a balance between some aspects in these patients.
Do type 1 diabetes patients have remaining stem cells that can be activated, or do they run out completely of insulin producing cells, or do they still keep some that just need to be regenerated?
I said before this, EVs derived from some cells like mesenchymal stem cells and other probable sources are one of the options on the table to regenerate and repair damaged or diseased tissues with lower risks and limitations. The investigation span a broad usage of EVs along with other materials. cells, tissues, biomaterials, or alone. In the last few years, numerous studies have been carried out in order to reveal the potential roles of EVs, especially exosomes, in tissue repair and regeneration. But their potential roles have not been fully elucidated. Some researchers propose approaches to increase the level of insulin-producing cells by some mechanisms and some pathways which may be transforming alpha cells, glucagon-making cells, to beta cells, which are insulin or insulin-producing cells. Beside this, the most important factor is modulating, suppressing immune cells that are destroying our insulin-producing cells. So the important factor here is doing these two functions beside each other, regeneration and immunomodulation.
How do EVs help with regeneration? Why do they help versus not using EVs?
As I told you, regeneration by MSC-derived extracellular vesicles, it's proved that inside the mesenchymal stromal cells-derived extracellular vesicles, there are some factors, some cytokines, some growth factors. These are like HGF, FGF1, IGF, VEGF, which facilitate the regeneration of insulin-producing cells inside the body. It's a little bit difficult to manage all of these actions in vivo. But in the laboratory and in vitro condition, it is probable, and different research groups around the world can manage these actions, and they can regenerate insulin-producing cells. But it's a little bit difficult in the body because balancing between modulating immune system cells and suppressing their attacks to insulin-producing cells inside the body is very difficult. And these hurdles a little bit are difficult and have some problems for researchers around the world.
Okay. Do you think that perhaps cancer uses immune-suppressing EVs so it can spread and function and avoid immune attack?
Yes, it's a fact nowadays. More research and more papers are published about this fact. They announced that, they revealed that cancer cells, totally tumor microenvironment, use some cells and some factors to produce some immune modulatory factors, like indolamin 2 and 3 deoxygenase, TSG6, or other factors, and they can modulate immune systems, and immune system cells don't attack the tumor microenvironment, and these cancerous cells can metastase to another parts of the body and can they can metastase to all of the other organs and promote their lives and their life cycle.
Okay. So in the near future, what do you envision that you will be able to figure out first? Are you running experiments with rats in labs and trying to restore pancreatic beta cell function? Or what is the specific focus of your research right now?
As a matter of fact, Considering newly proposed methods is an inevitable parameter in tissue, organ repair, and regeneration. Therefore, deep research is prerequisite for handling future studies. Regenerative medicine is a part of medicine that seeks the most applicable approaches to overcome some limitation in tissue remodeling, tissue regeneration, organ repair. I don't know. The future medicine will be attributed with the most facile recovery methods in personalized medicine, which are compatible, economic, and possess better regenerative effects. A major hurdle in the case of cell-based approaches toward regeneration is entrapment of injected cells inside some organ like lungs. These cells are somehow high risk due to their tumorigenesis. Accordingly, extracellular vesicles have been approved to be reliable substitutes instead of direct cell administration. It's still unclear which contents or properties of EVs are capable of promoting tissue regeneration. Future works are shedding the light on the exact possibilities of diminish any concerns, but our group is intended to cooperate more with any scientists and any research group from all over the world in different subjects. We hope that using EVs as biomarkers for disease prognosis and diagnosis, or using them as drug delivery vehicles, therapeutics to facilitate treatment of disease, especially type 1 diabetes, will be accepted, and EV therapy, as long as other procedures is applied.
Okay, very good. What is the best way for people to find out more and to contact you and get in touch?
Okay, you can anybody and people can contact me with my email addresses, mehdi.soleimanigl@gmail.com or soleimanim@royaninstitute.org, or in different social media like Instagram, Soleimani-GL, or I don't know, with Telegram, _Solei-G. These are my contact addresses that people can contact.
Okay. Well, Mehdi, thank you for coming. I know you're calling from a long way away in Iran, so thank you for being here.
Thank you very much. But is it possible to say final thoughts about the extracellular vesicles and things related?
Yeah, go ahead. We'll edit this in. Go ahead.
At last, but not least, by virtue of EV engineering, we might be able to modify the contents of EVs by adding, therapeutic drugs or compounds to enhance their regenerative potential in regenerative medicine. Moreover, seeking newly proposed strategies like combination therapies, for example, cell therapy beside extracellular vesicles, small molecule treatments besides extracellular vesicle therapy, and cell therapy besides small molecule treatments will pave the way of a new era in the future regenerative medicine. Thank you for your time and good luck.
Okay, Mehdi, thank you.
Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.